First chapter will be covering the concept of homeostasis. Now, if you open any book of physiology, the very first important chapter and concept that you will be seeing is homeostasis of the body fluid compartments, and that's what we'll be talking about over here. So let's get into the definition. First of all, homeostasis means the maintenance of constant conditions of the internal environment. This internal environment is also known as milieu interior. Now, before we go deep into this definition, let's just talk about the fluid compartments and this milieu interior, which it encompasses. So when we look at the human body, we are made of water. Without water, you're not going to live. So the average 70 kilogram adult male has got around 60% of his body made of water. So 60% of body weight is made of water. So that comes to around 42 liters. So your total body water in a 70 kilogram adult male, the average healthy person for his height and weight, would be around 42 liters. So you are made of 42 liters of water, okay, in the male. Females have a little lesser water because they have more subcutaneous adipose fat. We'll be talking about that in the respective chapter. But for now, just remember, total body water, written as TBW over here, refers to your total to around 42 liters. It is divided into two main compartments. One is ICF, intracellular fluid. Intracellular fluid is the fluid that's within your cells. Every cell in your body has water, so around 2/3 of your total body water comes inside your cells. So that comes to around 28 liters. 28 liters. All right? And this is present in all the cells of your body, from your neurons to your nephrons to your liver cells to your red cells and white cells and every other cell you could imagine. Then you have another compartment which is made of 1/3 of your total body water, the remaining 1/3. This is known as extracellular fluid, meaning it is fluid that is outside the cells, and this comes to around 14 liters. Okay, and if we look at the ECF, we can broadly divide it into two main categories. I divide it into three, but for your sake, I'm going to just say two categories. One is interstitial fluid compartment. Interstitial fluid compartment. This constitutes around 3/4 of your of your total ECF. 3/4 of the ECF. So 3/4 of ECF comes to around 11 liters, and this is the water that surrounds the cells, which is not in the vascular system. So it's ECF, and it's also not in the vascular compartment. And then we have the ECF, which is in the vascular compartment. This is around 1/4 of your total ECF, and we call this plasma. Plasma. And here your plasma has is around 3 liters. If you look at it, 3 to 3.5 liters in volume. Now, when you look at the term homeostasis, the maintenance of near constant conditions of the internal environment, also known as milieu interior, we are actually implying or talking about interstitial fluid compartment, though some books would just broadly classify it as ECF, or extracellular fluid. Now, this I'm going to just elaborate with another diagram over here. So this is your vascular compartment over here. Okay, your blood vessels and your heart, it's got the fluid inside it called blood. And if you see your blood, it's got two important components. One is the cellular components I'm going to draw over here, stacked over here. Around 40 to 45% of your blood is in the form of cells. It's composed of cells, cellular mass. 40% in females, 45% in males. We call that your hematocrit or even packed cell volume. So this is cellular constitution over here. Okay, red cells, white cells, and the cell fragments we call platelets. And then, so the water inside these cells that I've drawn over here, this is part of your intracellular fluid. And then you've got cells outside your vascular tree. These are your neurons, nephrons, hepatocytes, your cells in the retina, in your heart, myocardial cells in the, you name it. Any kind of cell can have this one, water over here. Now your ICF, intracellular fluid, is water from these cells and these cells put together. This is ICF, intracellular fluid. But now ECF it's composed of two compartments. I'm going to draw this in green. You see this fluid over here? This is ECF because it's not in the cells. However, this ECF, which is bathing your cells outside the vascular compartment, this is the interstitial fluid compartment we're talking about over here. This is interstitial fluid compartment. And if you look at the ECF in the vascular compartment, what I'm shading in red over here, that's plasma. Plasma. Right? So interstitial highlighted in green and plasma highlighted in red. This makes up your ECF. Now when we're talking about the constant conditions of the internal environment, we mean interstitial fluid compartments mostly. That's what we actually mean. Now this term called homeostasis was coined by an American physiologist called Walter B. Cannon. In 1929. And the term milieu interior, which is the internal environment that bathes your cells, was coined by a person called a French physiologist called Claude Bernard. And this was done much longer in the past, in 1865. So Claude Bernard wrote a book, and since I don't speak French, I'll just roughly translate it. It says that the fixed conditions of the internal environment, referring to the interstitial fluid, is absolutely necessary for life to thrive or flourish over here. So why are we so worried about maintenance of constancy of the internal environment, this constant internal environment conditions? Well, if you look at your cells, they are made of the building blocks of life called proteins, and if you are aware, proteins can change their structure in certain conditions. Like if you alter the pH of a certain protein or the temperature of a certain protein, the structure of a protein can change. Whenever you fried an egg or added vinegar to your food, you can automatically see that there are changes in the way how your food starts to look and even taste. That is because you are going to be changing the very structure of protein, and proteins are the building blocks of life. They make up enzymes, they make up transporters, pumps, carriers, structural proteins that lend structure to cells. So if you mess with proteins, your cells they can't function properly over here. That is why you need to maintain constant conditions of the internal environment. And since humans are open systems, that is to say, we exchange matter and energy with the external environment. So that means we can change the environment can influence our body temperature and also the composition of our fluids. We have to maintain a constant state of the internal environment for that reason. So because of this, when we speak about conditions, there are different variables or parameters that should be regulated. So the regulation of these variables or parameters by a system of control systems, by a group of control systems in our body, is necessary for life to take place, to thrive and to flourish. So what are these variables? Now, if we look at the open system, I told you matter and energy. So when we speak about matter, we are speaking about variables that are subject to variation. So let's take some of the simple variables we talk about. One is, let's say, sodium levels, potassium levels, bicarb, chloride. These are your ions that make up your blood. And there are many more: calcium, magnesium, you name it, selenium, zinc, everything has its role, copper. ion. So all of these are important ions. Then glucose, energy that exists in our fluid, ECF. These are different variables. And let's not forget pH. So hydrogen ion concentration is also very important over here. That means pH, okay, the negative log of the hydrogen ion concentration. And then we have temperature. So if it's too hot, naturally your proteins will maybe function a little too much and then start to get denatured over there. And if it gets too cold, sometimes your proteins, which may be enzymes, might stop working so much and eventually also get denatured and lose their function. Then you also have another important thing. So the volume of these fluid compartments, it's also very important to regulate this. The volume can translate to pressure also in some cases. So these are your different parameters over here. Ions, you even have gases like oxygen, CO2, and all of this. And there are many more. And the body tries to maintain a fixed type of internal environment. And how is this possible? This is because nature has created our body such that we have certain set points. This is an important thing. Set points or reference ranges. Now these set points or reference ranges are determined by nature, and it is what these variables have to be maintained within or around a certain value. Like for example, the pH of your blood is somewhere around 7.35 to 7.45, and this is the range. There's a little bit of freedom of movement within this range. As long as the pH of your fluid compartment is within this range, it's fine. Nothing bad's going to happen, hopefully. And if we look at other ions and other parameters like this, let's say glucose, it hovers around 100 milligrams per deciliter in its value. All right. Sodium is around 123. 135 to 140. 135 to 140. So let's say 140 milli equivalents per liter. So we have these various parameters over here that must be regulated. If we look at temperature, 37 degrees Celsius. But of course the range is 36.6 to 37.2 degrees Celsius. So again, ranges are there for every parameter in physiology, and they have certain set points, and we must maintain those parameters in a certain range because these parameters are subject to variation. So how does the body maintain this constancy of the internal environment? It's because we have certain systems in place. So these systems are located in our body and they have a certain goal. So the goal of the system is to maintain the set point within a certain range. At least that's what these things are. So systems are a sort of setup which has the goal of maintaining the variable within a reference range, within the set point. So this is a system. Now, how do we classify these systems? Well, we have two types of systems. One is the feedback systems. Feedback system. And then you have the feed-forward system. Feed-forward system. Now, what is this feedback system and feed-forward system? So again, feedback is further divided into negative feedback. This is very important and very important for homeostasis. Negative feedback. And we also have positive feedback over here. Positive feedback. Now, let's talk about each of these in detail. So a feedback system gains information. It takes information, it senses information, and then it makes a decision and it gives back a response in order to control this variable. So let's give an example. So all these systems, what we talk about over here, they have something called a sensor that senses the variable. Then it has these sensors are going to go in the form of a signal. Now, those signals could either be in the form of electrical signals called action potentials or membrane potentials, or it could be in the form of chemicals that are released in response to, or in corresponding to a certain level of some parameter that we're looking at. And this sensor is going to go to an integrating system. So the integrating system is sort of like a chip, a computer chip, which has a sort of algorithm in it. And this algorithm is such that if there it has the set point or the reference range in it, in this chip, and depending on how much the value, the amount of variable is sensed, variable or parameter is sensed, this sensor is going to give a signal to the integrating system, and that integrating system is going to compare this signal strength with the set point. So there is a strength of the signal based on how much of this and a deviation, or how much a level of a variable has deviated from a set point, and that strength of the signal goes to the integrating system, and this integrating system is going to compare that signal with the set point or the reference range, and it's going to say, okay, too much or too little, or is it within the right amount. So it's sort of like, is it too much or is it too little, or is it just right, like Goldilocks. So here the set point, if the variable comes, the value of the variable is within the set point, then the integrating system says, okay, everything's going nicely, let's just keep things going on how they are. But if there is a variation and there is, let's say, a drop in a parameter, in a value of a parameter, for example, your sugar levels, if you don't wake up on time and don't eat breakfast on time, and you start going for a jog in the morning, skipping your breakfast, it's very likely that your sugar levels will drop and you will feel a little exhausted and tired as the day progresses. So the sensors could pick up these levels of the sugar level, and it will try to say, okay, the sugar levels are getting low, I should do something to raise the sugar level, as an example. So then a signal is given to an effector organ. So this effector signal is going to activate an effector organ, and that is going to give a response that tries to oppose the change and restore balance, bring the parameter back to the set point, and hence maintain homeostasis too. So to summarize this little concept over here, so the variables that are subject to change, these parameters that are measurable over there in our body, that make up your milieu interior of the cells, they are sensed by sensors. The level of this variable is sensed by a sensor, and the signal goes to the integrating system, and the integrating system is going to decide, is this within the reference range or the set point or not. And if there is a variation, then it's going, the integrating system is going to give a signal, an effector signal, based on how much the variation has taken place over there. And then finally you get a response for that. Now this is a very simple simplified version of this explanation, but let's go into a little more detail.